Yeah, thanks, Uli, and thanks for the invitation to speak about some of our recent work in our lab in trying to generate phospholipids and phospholipid membranes, and using enzyme-free chemical methods. So we all know that lipid membranes are essential to life. Lipid membranes literally define the boundaries of extant life. And so I think there's a lot of reason to believe that lipid membranes were present during the origin of life, and many theories on the origin of life invoke various types of lipid membranes as compartments, which are, of course, really important for evolution. But when we think about lipid membranes in current life, and we know that lipid membranes, they can promote and protect chemical reactions, they can control the exchange of matter with the environment, they can define boundaries, and very important and often not as much talk about, we had some nice talks about this before, is that they can maintain ion and pH gradients. But I think one of the things that we've always recognized, it seems like the lipid membranes are perhaps understudied compared to work that's being done on, for example, proteins and nucleic acids. And so one of the things we're really interested in is how do we develop ways to mimic the function of lipid membranes using chemical reactions. And if we think about cells themselves, well, true living cells today, lipid membranes are extraordinarily dynamic systems. They're constantly undergoing synthesis and degradation and trafficking, and they're basically far from equilibrium active matter. And this kind of contrasts a little bit when we start thinking about the typical ways in which we sort of use lipid membranes in the lab. Oftentimes, and we've done this in our own lab as well, in studies of synthetic cells, we sort of use a lipid as a compartment, and that's it, sort of a very static compartment. And I think that's very different than from how lipids behave in real life. And so I think one of our goals in our lab is to try to develop chemical reactions in a way to sort of mimic the more dynamic, far from equilibrium nature of lipid membranes. And so the idea of mimicking lipid membranes is not an entirely new idea. In fact, many years ago, the late Janos Fendler came up with a really nice term, membrane mimetic chemistry, and he wrote a really nice CNE News review article about this, if you're interested, back in 1984. And the idea was, you want to create systems that mimic aspects of biomembranes. Interestingly, he was really focused on practical applications, so controlling the rates and stereochemistry of organic reactions, enhancing solar energy conversion, or targeted drug delivery, which of course is very, I think, prescient of him, because of course, most of us now have received vaccines from Pfizer or Moderna that used lipid nanoparticles, and clearly now lipid membranes are extremely important in drug delivery. But interestingly, at the time, Fendler really kind of went out of his way to point out that, look, membrane mimetic chemistry is not actually about literally mimicking the function of biological membranes. That's not the real claim. And perhaps maybe at that time, it was really sort of challenging to imagine, well, how are you going to mimic all the, growth, reproduction, transport properties of living cell membranes? But, moving forward, I think in the current time with, advances in molecular biology and in organic synthesis and in the analytical techniques we have, I think we can revisit this term of membrane mimetic chemistry and expand it so that we actually really go about trying to mimic, the types of functions that membranes have in cells. And this, of course, tails really nicely into what we're trying to do as part of the origin of life as well, as we want to create systems that mimic life. And so, can we create membranes that can grow, or can we try to build in transport or maintain ion gradients, or can we look at sort of the fission of protocells, and can we also make different kinds of membrane phases that mimic the function of organelles? And if you're interested in this topic, one of my graduate students, Jacob Vance, wrote a really nice perspective recently, and I direct you to that. So, in our lab, when we first started, we started thinking about ways in which we could use chemistry to mimic lipid synthesis and, in membrane formation. If we think about the conventional ways we make membranes, well, very typical way is to, take a dried film of phospholipid and add water, and you hydrate it, and you form vesicles. There are more sophisticated techniques that use, microfluidics or inverse emulsions to form your vesicles. But in both these cases, you're always using preformed phospholipids or lipids. And in biology, of course, cells are making these lipids synthetically and coupling that synthesis with the self-assembly. And so in thinking more about this, it's also it's also good to think a little bit about, why are, in at least modern biology, why are phospholipids such a privileged class of molecules for making membranes? And, and many in this audience probably know this, but basically one of the very simple ways of explaining it is the shape of the lipid. So phospholipid, if you look at the head group and the two hydrophobic tails, the cross-sectional area is roughly similar. And so you can think of the shape as that of a cylinder, and that cylinder is going to want to pack to form a bilayer. In the case of we remove one of these hydrophobic chains, well, now you're going to have a conically shaped molecule, wedge shaped, if you will, and that's going to want to pack to form a micelle instead. So I'm thinking more about the sort of difference between sort of the two chain versus one chain lipid species. We got attracted to thinking about mimicking a pathway in modern biochemistry in which you have two single chain precursors forming a double chain membrane forming product. And this is the so-called land cycle or remodeling pathway in phospholipid synthesis. You actually have phospholipids that are cleaved, one of the acyl chains is cleaved by a phospholipase, and that lysophospholipid is reacted with a thioester derivative of fatty acid through the action of an acyl transferase. And so you have these two single chain precursors, and in a single coupling step catalyzed by the single enzyme, you can form now a two chain membrane forming product. And so we thought that's a really, elegant way to go from non-membrane forming precursors to membrane forming product. Maybe we could try to mimic this step now, not enzymatically, but non-enzymatically using organic chemistry. And so again, that's really the simple idea here is you take two single chain precursors, use chemoselective chemistry to make now a double chain product, and that's going to lead to a compound that can self-assemble and form membranes. And over the years, we've done this now with several types of chemistry. For example, click chemistry with azides and alkynes, negative chemical ligation, which some of you might be familiar with in peptide chemistry. We've sort of utilized it instead to form phospholipids, and esterification reactions as well, which is what I'm going to talk about in a little bit. And in all these cases, we can actually watch the de novo formation of membranes. So we can have situations where we have no pre-existing membranes, we mix together the precursors, and you can form these membranes and visualize this using fluorescence microscopy and membrane staining dyes. And because these reactions are really efficient, we can also actually watch phenomenon like membrane growth, or even curvature changes, even sometimes primitive fission in these membrane systems. Very heterogeneous, not really as clean as, for example, cell division, but we can still see things like the blebbing, for example, of membranes off of an initial vesicle. So, why are these reactions? We've many times noticed these reactions are really, really rapid. The formation seems to sometimes be better than what you would expect if these reactions were just between soluble species. So what do we think is actually happening? Why do we think the reactions are so efficient? So if you remember, these precursors are single chain precursors. And so they have a tendency then to want to self-assemble to form micelles. And these are essentially now reactive precursors. So once they form the micelles, they're going to react with one another. And they're going to be in close proximity to one another. So the reactions are going to be really, really fast, very efficient. And as you start converting the single chain product to the single chain precursor to the double chain product, you're going to start forming membranes. And then these membranes can form, vesicles can spontaneously emerge, and you can get more product to be fed in, and these vesicles can potentially grow as well. So this is at least one thought. Obviously, this is a cartoon that we've drawn up as to how this sort of process is occurring. So, for many years, we basically worked with very abiological phospholipids. We made phospholipids, they could form membranes, they kind of approximated natural phospholipids, but they always had some kind of unnatural linkage, like a triazole or a cysteine, like an amino acid inserted in there. And it always occurred to us, well, it'd be really interesting if we could develop ways to generate natural phospholipids, diacyl phospholipids that are identical to the kinds of phospholipids we find in modern biology, but to do this non-enzymatically. And, we tried some stuff after working with thioesters. We tried, taking lysophospholipid and reacting it with a thioester derivative of a fatty acid like oleic acid. And, if you do this with a couple of different thioesters, you get, trace, if any real conversion, no membrane formation whatsoever. And so a really talented organic chemist, Luping Liu, who actually did her PhD with Ben List, who probably recently won the Nobel Prize. Luping, a very, very talented organic chemist, decided to join our lab as a postdoc. And she looked at this problem, and I think it's what's really great about having some fresh eyes look at these problems. She said, well, you looked at neutral species and you looked at negatively charged species. Why not look at a positively charged species? And so she made a thioester from a very simple thiol, thiocholine. And when she made that thioester with the oleic acid, now when you mix these two together in mildly alkaline solutions, you get a near quantitative yield of the natural phospholipid. And so this is kind of a, to us at least, a really remarkable finding. And so, why does this work? One thing that, we thought about, or what Lu Ping speculated, is potentially the positive charge on the choline is ion pairing with the negative charge on the phosphate ester of the lysophospholipid. And we did some DFT calculations with Ken Hoke at UCLA that seemed to indicate that that is at least one of the reasons why this reaction is working so well. Nevertheless, at sub-millimolar concentrations, you can mix together these single chain species. They form presumably mixed micelles. You have then the acylation reaction, and you can form these natural phospholipid species. And this can be done at, even in sodium bicarbonate buffer, pH roughly around 9. And you can watch the de novo formation of, cell-like lipid membranes. This can be done with different kinds of head groups. It can be done with different kinds of acyl chains, different lengths of acyl chain of fatty acids derived thioesters. But the length is important in the following way, that you do need to be, in terms of your concentrations, above the CMC of these single chain precursors. If you're below the CMC, you don't get the reaction to occur very well. And so again, that lends itself to this idea that basically these precursors are assembling and forming these micelles, and that's within the micelles, that's where you're having the reaction take place. So one of the questions we had since we're working in mildly alkaline solutions was whether or not this reaction could work in mildly alkaline solutions from natural water sources. So there are water sources on the planet that are basic, essentially, and we were curious, whether or not this chemistry could occur spontaneously from those kinds of sources. So one such source are the soda lakes. So there's many soda lakes around the world. There's been some really interesting recent work suggesting that soda lakes may have been a privileged environment for the origin of life. So in California, we have a soda lake called Mono Lake that's about 7 or 8 hours, I think, drive from San Diego. And I was kind of chatting with Lu Ping one day about this idea of, it would be interesting if this could work in soda lake water. And then, 2 days later, she emails me and says, oh, it works. And I said, what did you do? So it turns out she got so excited by this idea, she hopped in the car the very next morning, very, very early in the morning, drove all the way up to Mono Lake, collected water, and then went back down to San Diego, filtered it, and then ran this reaction. And sure enough, you could get a high yield of the phospholipid, and you can visualize vesicles, in this case by transition electron microscopy. Another environment that's thought to be a potentially privileged environment for the origin of life, there's been a lot of discussion about this, are the alkaline hydrothermal vents, particularly the Lost City hydrothermal vent field. And this has been work that's been done by Mike Russell and Bill Martin quite a bit. And these are vents that spew out alkaline fluid. It's warm, 49 degrees Celsius. There's also various dissolved gases and organics potentially. And so we were curious whether or not a reaction could work in hydrothermal vent fluid. Now, getting hydrothermal vent fluid is not as easy as driving up to some place and taking a sample. These vents are located thousands of feet below the ocean in the middle of the Atlantic Ocean. And so we decided to collaborate with Susan Lang at the University of South Carolina, who does these really cool NSF-funded missions where they take these remote operatable vehicles and submerge them, and then they collect the vent fluid from the Lost City hydrothermal vent field, which is what you're seeing here in this video that she provided me. And so we were able to get these vent fluid samples from Susan, and sure enough, we were able to show, in fact, that our reactions work in the vent fluid, and you can form really nice giant vesicles from these precursors. So, we're making these vesicles now in alkaline media. And so reasons to believe that in the lumen of these vesicles, you're going to also have, a basic pH. And so one of the things we wondered about, and this is really kind of inspired by the work of Russell, is the thinking that, well, if you're forming these vesicles in the basic pH, what happens if you switch the outside environment to something that's more acidic? Could these phospholipids that have spontaneously formed non-enzymatically, these membranes, maintain a pH gradient? And so, we went ahead and encapsulated a pH-sensitive dye, HPTS, inside these vesicles. If this dye is exposed to something more acidic like citrate buffer, you see almost no fluorescence whatsoever. But if you put it now in Mono Lake water, which is at pH 10, now you have a very, very high fluorescence. And so the idea was, well, what we'll do is we'll make these vesicles now in the Mono Lake water, and then we'll do a buffer exchange from the outside to a citrate buffer. And if there's a lot of leakage, well, the HPTS is just going to be quenched. But if there's no leakage, or if there's some ability to maintain this pH gradient, we're going to be able to see this fluorescence, and it's going to maybe decay slowly with time. And so Looping did this experiment, and actually when you take these vesicles and do the buffer exchange, you can see the inside is extremely bright. So these are vesicles made now spontaneously, non-enzymatically, there's de novo vesicle synthesis in the Mono Lake water. We've done the buffer exchange, citrate buffer, and immediately you can see there's quite a bit of fluorescence. And this does decay, so after 2 hours, it gets less bright, but you can still see there's fluorescence. And then after many hours, 15 hours, the fluorescence is much lower. So it's pretty interesting to think that you can now, in this sort of spontaneous way, create these membranes that can maintain a pH gradient. And there's this idea out there that perhaps these vents are very interesting because it's thought that the Hadean Ocean was more acidic, and the vents were basic. And so now, if you have a membrane that's separating the more acidic hydrothermal vent water from the acidic ocean, that you could basically get a pH gradient for free. And this might be one way in which you could drive the evolution of molecular machines that can harness that pH gradient for some sort of purpose. So with that, I'll wrap up, and I'll just want to thank my team, really dedicated group of chemists and biochemists that are quite literally interested in understanding the interface of chemistry and biology. And again, I want to thank the organizers, and I'm happy to answer any questions.